wonderful to be able to be here and see the people who are affected by our work, and to get this crystal. I have a prior one which got slightly damaged in the earthquake, but it's so I have a set now, so that's great. So I'd like to get right into it. I realize most of you have no particular background in neuroscience, so I'd like to start with some basics and try and explain what we know about narcolepsy, what we don't know, and also something about what the normal function of the hypocretin cells, the cells which are missing in narcolepsy, what they normally do, which helps us understand what happens when they're not present. So the brain is composed of neurons, nerve cells, and other support cells called glia. I won't be talking about them. Neurons basically release chemicals onto other neurons. That's how the brain functions, and it's this processing of chemical inputs to neurons that eventually produces behavior, that produces sleep and waking states. So this is a schematic of a neuron. You see at the left in the top corner is the dendrites, which is where the input comes into a neuron, and then on the right there's the output goes along the axon, that's the output of a neuron, and it ends in these little terminal buttons which is where the transmitter is released onto the dendrite of the next cell. In the lower right corner you see how this release occurs. It occurs from little packets within the tip of the axon. They kind of burst open and the molecules are deposited on the dendrite, which then responds, and the chain continues. This is how information is processed. There are more than 100 types of neurons in the brain that have been identified, different types meaning cells that use different transmitters that release different chemicals at their terminals. There's probably a lot more than 100, but I'm just being conservative in saying 100 types have been identified. So the one that has been of most interest with regard to narcolepsy is hypocretin. So I thought you'd be interested in sort of the math here. There have been counts of the number of neurons in a human brain, and the general estimate is about 100 billion neurons in the human brain. Some people say 80 billion, 100 billion, but the 100 billion is a nice round number. Of these, only about 76,000 use hypocretin. Again, this is the chemical that's implicated in human narcolepsy. So if you do the math here, that's 0.00076%, or less than 1 in a million neurons in the brain contain hypocretin. Yet this very small population is responsible for the illness that many of you suffer from, the loss of a portion of this population. So in our study in 2000, we found that about 90%, on average, 90% of these hypocretin neurons are missing in people with narcolepsy. In a subsequent paper, we found that about a third of these cells are missing in narcolepsy without cataplexy. And these findings require analysis of human brain tissue, of normal human brain tissue, and the brain tissue of people with narcolepsy. So I can't resist a little commercial here. That we're dependent on people willing their brains. There are brain banks. We work with an NIH brain bank in Los Angeles, and if any of you want to will your brain for research, I can contact you. You can contact me. I can give you the information. And one caution is, my experience is that usually it's people in their 20s and 30s that are eager to do this. And since I'm planning to stop doing research before I'm 100, unless you're in a motorcycle gang or anyway, obviously this is critical to understanding narcolepsy, and I really am grateful to people who have made this donation in the past. I don't know how many of you know Sue Corella, who died last year, and she willed her brain, and we're working on it right now. So the finding of loss of hypocretin cells in humans has to be considered in light of the animal models that also exist. We know that if you genetically eliminate hypocretin cells, you get all of the major symptoms of narcolepsy. And we know that there's a loss of hypocretin cells in people with narcolepsy. So it's quite clear that the loss of hypocretin is the cause of narcolepsy. Perhaps there are some rare cases where that's not true, but in the vast majority of cases, that's quite clearly the case. So I thought I would show this to show you where we're talking about. The hypothalamus, you see that? Can you get the lights down a bit? That might be helpful. The hypocretin cells are in the hypothalamus. The hypothalamus is right in the center of the head, really. And it's within that little red circle that I've indicated on this section of the human head. It's right above the pituitary gland. It's right behind the optic chiasm, which is where the optic nerves cross. Even within the hypothalamus, the percentage of cells that contain hypocretin is way less than 1 in 1,000. So it's a very small group, and as Dr. Thorpe mentioned, narcolepsy really is unique among neurological disorders in how specific and subtle the underlying deficit seems to be. So 1/1,000,000th of the cells in the brain are affected. And, you have to compare this to other degenerative diseases, which in some cases 10, 20, 30% of the cells in the brain are lost. So it's very hopeful, too, because if any neurological disease can be cured or reversed, narcolepsy would be the one. And this is something we're certainly hoping to do, and others are working on as well. The new development concerns another type of cell that's also in the hypothalamus, and these are cells that use histamine as a transmitter, so hypocretin and histamine. Histamine also helps maintain waking. We've known that from basic science studies for many years. And you all have probably experienced that when you take antihistamines, you get sleepy because it's blocking these histamine cells in the brain. But as you may also know, newer antihistamines do not act in the brain, so they don't make you sleepy. That's one of their advantages. But if the histamine cells in the brain are inhibited, it definitely causes sleepiness. So the new finding is that the number of brain histamine cells is increased, greatly increased, in the brains of people with narcolepsy. So just to back up a bit, this is what we saw in the hypocretin system. So on the left side is a cross-section of the hypothalamus that I showed you in the human skull. In the center on the left shows the distribution of the hypocretin cells in the control individual. And on the right side, you see the depletion of these hypocretin cells in a person with narcolepsy. And the cells that remain look relatively normal, but they're only 10% of the baseline level. In our studies, we always see remaining cells, remaining hypocretin cells. Now, this is the new finding. And here you see in red on the right a plot of the histamine cells in narcolepsy, and on the left, the histamine cells in a control. And you see there's a greatly increased number. When you do a count, you see that on average, the people with narcolepsy have 65% more than our control population. 65% more. So this is a plot of the subjects we studied. The controls are in blue, and the people with narcolepsy are in red, and they're plotted according to the number of cells. And you see there's no overlap whatsoever, that the number is higher in all of the narcoleptics than in any of the controls. When you look at the subject all the way to the right, this individual has more than double the average number. So there's a tremendous increase. And I should point out, Dr. Scammell will be speaking here, I understand, later. He has the same finding, and both studies are now online, but they'll be sort of officially published back-to-back in Annals of Neurology. So this is a simultaneous finding by two groups, which I think can give you great confidence that this is real. There are some minor differences between our findings that I won't get into, but the finding in the human is exactly the same. Now, what we see, and this actually is the only difference between our study and Dr. Scammell's study, is that we don't see this increase in histamine cells in the animal models of narcolepsy, even though these animal models show all of the symptoms of human narcolepsy. The basic symptoms are sleepy, they have cataplexy. And we looked at 4 different models. We spent a lot of time on this, and we were very puzzled that we didn't see it, but we're quite confident that it's not there. And clearly, so we don't need this loss, this increase to have the symptoms of narcolepsy in the animals, and clearly having the histamine cells you would think would increase alertness, but obviously it does not, sort of by definition. So I thought Dr. Thorpe made a good joke in saying that I would explain how this causes a symptom. What we know is that this is as big a change as we see with hypocretin. So this is a huge change, and we don't understand it, because despite the fact that the histamine cell number is increased, if you actually measure histamine in narcolepsy, it's abnormally low. So it may be that part of what's going on is the histamine is not being released, it's building up in the cells. We need to look at more of these cells and do more analyses to see exactly what's going on. But one thought that occurred to us is that the reason you take antihistamines is because the histamine in the periphery causes inflammation, causes allergies, and you take antihistamines to prevent that. Histamine in the periphery is part of the immune system. It's part of the mechanism for killing foreign cells. And so we wonder, without any particular evidence here, but the difference between the human narcolepsy and the animal narcolepsy is the human narcolepsy we know is produced by autoimmune processes, as Dr. Thorpe just mentioned. And we wonder whether the histamine, the abnormal increase in histamine, may be related to what has happened 40, 50 years before these patients died, that the histamine may actually have killed the hypocretin cells. So that's a speculation at this point. We continue to investigate it. And also the fact that there's this increased number of histamine cells suggests that there might be some way of expressing more histamine in people with narcolepsy as a treatment, because this, we know, is a natural alerting chemical. Now, the other thing which is quite amazing about this and really important for the field of neuroscience is this is really the first evidence that I know of an increase in the number of neurons in any brain system, in any disease in humans. So you have this 65% increase in the number of neurons. Now, the conventional wisdom, I have some allergies myself, so. The conventional wisdom is you're born with a certain number of cells. At some point in development, they're created, and then you keep that number for the rest of your life. And there's a slight decrease with aging. And then in certain neurological diseases, there's a massive decrease with aging or with disease onset. But there is no example of an increase in a particular number of cells, a cell type, in any human disease. And I would say this is the first evidence that such an increase occurs in any mammal under any conditions. Now, there is neurogenesis in two parts of the brain that's been studied, a region called the hippocampus and the olfactory bulb. The dentate gyrus is part of the hippocampus. And in these regions, there appears to be turnover of cells. So the new cells are formed to replace the old cells. But there's no evidence of such turnover in the hypothalamus. And yet we're seeing this huge increase in the number of cells. So just to speculate further, the fact that you can increase the number of cells. Now, we don't know what did it. And that's something we're trying to understand, but it suggests a very exciting possibility that perhaps we could increase the number of hypocretin cells. And that's something we're very interested in. If it's possible to increase the number of histamine cells, how do we go about increasing the number of hypocretin cells? Now, one way of doing it is to surgically insert genes to produce new hypocretin cells. This is an approach that's been tried in Parkinson's disease for many years, with mixed results, because when you insert these cells, they don't have the appropriate connections and they don't have the appropriate control. So they can cause abnormal function, and that then essentially becomes irreversible. But if you could increase the number through some more or less natural process, and obviously that's occurred in the case of histamine, this would really be big news. And so I'll just leave it there, but for me, this is the most exciting aspect here, that this has great implications for potential treatment of narcolepsy and other neurological disorders. So just to back up a bit here, Dr. Thorpe already went over this, the evidence that narcolepsy is an autoimmune disease. And in our initial paper in 2000, we saw evidence of what's called gliosis, a proliferation of support cells in the brain. These are the other cells that aren't part of the 100 billion. These are cells that are created or proliferate from inflammation, among other things. And we saw in our very initial studies, very first study, that in people with narcolepsy, there's a proliferation of glia in the region of the hypothalamus where these cells are lost. So that's certainly consistent with the circumstantial evidence from influenza and from strep and from this HLA linkage that Dr. Thorpe mentioned. It all fits together, and it's quite clear that there's an inflammatory process in the brain triggered by strep or other kinds of infection or H1N1 that then causes these cells to die, or perhaps causes the histamine cells to kill these cells. Now, in 2000, we tried just injecting hypocretin into narcoleptic dogs, which we have. And in fact, we saw with intravenous injection a reduction in cataplexy and a reduction in sleepiness. The narcoleptic dog has a receptor deficit, not a loss of hypocretin cells, so it's a little different from the human condition. Working with Sam Deadweiler at Wake Forest University, we studied the effect of intravenous administration of hypocretin and also nasal administration. Nasal administration gets peptides into the brain much more rapidly than intravenous injection because it can go through the olfactory mucosa and into the cerebrospinal fluid. And what Deadweiler did was study monkeys. These are normal monkeys, but they're doing a task that's sensitive to sleep deprivation. So it's called a delayed matching sample task. The monkey sees something, and then after some delay, it sees three things. It has to choose the thing that it saw before, and then it'll get rewarded. And so this is very sensitive, as you might imagine, to sleep deprivation. And what we're seeing here is that if you give Orexin, which is what's labeled here, of course Orexin and hypocretin are the same. If you give hypocretin, these animals, their performance returns to baseline levels after sleep deprivation. And if you actually look at brain metabolism in these monkeys, you can see that the brain metabolism normalizes. So this is something called a PET scan, which measures the metabolism of the brain, and in the left column you're seeing blue, which indicates areas of hypometabolism, and at the very bottom an area of hypermetabolism. And it's not really necessary to get at the details here, but you can see that the colors change when you give hypocretin. You're activating the regions that had been deactivated, and you're deactivating the regions that were abnormally activated in the sleep deprivation condition. So unfortunately, this approach has not been tested very much in humans. There's not a lot of money in it for drug companies, and it's very expensive to do this testing. But there is some work going on giving it to humans with some positive results. You would think that hypocretin administration would be the ideal treatment because it's replacing what's missing, although obviously you can't, by inhalation, necessarily get it to all the places that it would normally be released in, but that's also true for Xyrem or amphetamine and any other drug systemically. But this is the chemical that's missing, so you would expect that it's going to be more effective. So now I'd just like to talk about what the normal role of hypocretin is as a way of understanding the deficit that you get when it's lost. Now, the other name for hypocretin is Orexin, and this name was coined by the group led by Yanagisawa, which was the second group to identify this peptide. So hypocretin and Orexin are exactly the same thing, if you're at all confused about that. But the speculation and the evidence presented in the initial papers indicated that it had something to do with feeding and that you would be anorexic, which is why they called it Orexin. You'd be anorexic if it was missing, so that this was an appetite-stimulating chemical. So we tried to directly test this hypothesis, and this is in normal dogs. We have narcoleptic dogs, and we have breed-matched control dogs. And we food deprive these dogs. They normally get fed once a day, so 48 hours food deprivation is actually just missing one meal, but they're hungry. And we looked at the level of hypocretin in the cerebrospinal fluid. And instead of going up, which is what you would expect for an appetite-stimulating chemical, it actually went down, but not significantly. And then you see these other points on the plot show that nothing much is happening, but to the extent that anything's happening, it's completely inconsistent with the idea that hypocretin stimulates the appetite. And the other factor that's consistent with this is that there have been several studies showing that people with narcolepsy are more likely to be overweight. This is a little hard to interpret because obviously most people with narcolepsy have been treated with a variety of drugs, and this could induce that. But now that we have these animal models, we have a perfectly controlled situation where no drugs have been given. And in these animal models, we also see increased body weight, increased fat in the animals with deletion of this very small cell population. So I think it's pretty clear that that feeding control is not the main issue here, or at least not stimulating feeding. Now we have, our dogs are in a colony. They're in relatively large cages, but every day they get to go out in this big grassy area and play, dig holes, fight with each other, and have a good old time. And so we sampled, you know, I was thinking initially that hypocretin might be related to activity. And these dogs are very active. And indeed, when they're out in the yard, the hypocretin level goes way up. In contrast to what we saw with food, where there wasn't much of an effect, you can get a 70% increase in hypocretin level by just letting these dogs out in the yard. So I thought that's a rather crude way to present science. And if it's activity, then maybe I can just get these dogs to run on a treadmill. And there are commercial treadmills for dogs that are advertised as your doggy will love running on this treadmill. And so we got one of them. But we found that our doggies that were used to going out in the yard weren't too crazy about running on the treadmill. But they did do it, and they did it very vigorously, and they would run for an hour, with breaks thrown in, and their body temperature would go up just as it did in the yard. Their blood pressure would go up just as it did in the yard. Their respiration rate would go up just as it did in the yard. And this is what we saw with the hypocretin level. Absolutely nothing. So this is very interesting because it rules out all those other variables. It's not related to heart rate, not related to blood pressure. I mean, it may be related to, but you can have all these changes without any change in hypocretin level. So as I've implied, when we got these results, we were furious. But then thinking about it, what's the difference between running on a treadmill and running in the yard? And as I've implied, they like running in the yard, and they interact with each other, and maybe they like running on the treadmill a little bit, but it's obviously not their favorite thing to do. So we've been investigating this emotional aspect. And of course, we know that cataplexy and narcolepsy is related to emotional stimulation, so we realized that was kind of a fit. And so in this study, we're looking at these mice that have deletion of the gene that produces hypocretin. So these are called knockouts. So you may see KO in some of these slides. So these are essentially narcoleptic mice. And we compared, and you can actually do these with littermate mice. So some will have this mutation and some will not. So they're very closely related, except for this loss of hypocretin. And we trained them on a task that involves bar pressing for food. I'll show you this. Each bar press, each subsequent pellet requires a little bit more work. So one bar press gives them one pellet, then the second pellet they got to do 2, the third pellet they got to do 3, etc. So this is a way of measuring sort of the maximal extent of their motivation. On the right, you see what the normal littermate did. It bar pressed 3,900 times in 2 hours. Our champion did 10,000 in 2 hours. So you can do the math there. It's quite incredible. They pressed very well. But the knockouts were never able to complete the 2-hour period. So you see on your left, the horizontal line means they stopped bar pressing. And if they stopped bar pressing for 15 minutes, we terminated the session. No knockout ever completed the session. So they just stopped. And it's not just related to food, because when we had them bar pressing for water, we had exactly the same result. So it's related to reward. And so on the left side here, you see just that it's significant. There was a significantly lower bar press rate in the knockout, which is to say the narcoleptic, than in the wild type, which is our shorthand for the normal mouse. But we trained them in another task, which is sort of an avoidance task. So they're on these metal bars, and we give them a mild irritating foot shock. We're not electrocuting them, but we train them to bar press to avoid this foot shock, and so they have to bar press in the same way to avoid it. In that situation, the knockouts had no problem at all. They worked just like the normals. And so this is a video of 2 mice. Now, 10 minutes into the session, which is to say at the beginning of the session, they're both bar pressing just fine. So the narcoleptic one is on the right, the wild type's on the left. They bar press and then they go over to the bottom where the food is to collect their winnings. So at the beginning, they both start off fast, as you saw on the graph. There's no difference. There's no difference in the speed of learning the task either. But an hour later, you see the wild type is bar pressing away, and the knockout has fallen asleep on the lever. You can see it does move at one point, proving it's not still. Okay, there it goes. So the other interesting thing here is that in the dark phase, the knockout has no problem with the same task. So the deficit is in the light phase, not in the dark phase, and I'm sure you all experienced being unable to sleep. The trouble is for humans, it's the light phase that we're trying to perform in, not the dark phase. I think the hypocretin is related to the arousing effects of light in both humans and in mice. In mice, it's not so important because they're normally asleep. They don't miss it. But in humans, it's very important. So there's another technique that we've used, which is called immunohistochemistry. It's a way of looking at the brain of an animal that has been in a task and seeing which cells have been active during that task. So this is done in the normal mice, the wild type mice. And what we see, so you can see the red arrow at the upper right is the phos stains the nucleus of the cell. So you see the black dot in the center that the red arrow is pointing to, and the hypocretin is staining the rest of the cell, the cell body. And so if you see a nucleus labeled within the cell, that means that cell has been very active within the last 2 hours. And so in red, we see here in the light phase hypocretin neurons that have been active during the task. So you see a lot of red dots on the left on the bottom. That means that in the light phase, all these hypocretin neurons were active during the 2-hour period when they were performing this task. But in the dark phase, they're not active at all. And this is very paradoxical because the dark phase is when mice are normally active. So they're more alert, and yet the hypocretin cells don't seem to be participating. They're only concerned about arousal during the light phase. And so this perfectly explains the deficit we see in the knockout, that In the wild type, they're only involved in the light phase, and when you remove them, the deficit is only in the light phase. And so through the wonders of Medline, I just happened to search for light and narcolepsy, and there is indeed this paper in 1989 that compared the effects of light activation in people with narcolepsy with people with hypersomnia from other causes. And people with hypersomnia are activated by light, but people with narcolepsy are not activated by light. So really exactly what we're seeing in these mouse models, it all seems to fit together very well. And so this summarizes some of these results. You have this, at the left you have this high level of activation of these cells, bar pressing for food in the light, low level in the dark, which is really basically baseline level, low level during shock avoidance, and low level during chamber control. So shock avoidance, obviously, that should be anxiety-provoking and highly arousing, and yet these cells are just not involved in that. So they're involved in a particular kind of arousal for pleasure. Now, this is an example of a neuron. So we've developed techniques to record from these neurons in rats, and I hope you're not offended by seeing an implanted rat. The rat's in absolutely no pain. I will show you this video. This shows what happens. We have an animal that's asleep at the beginning, and we give him a piece of chicken. Now, that wakes him up, and so you see the EEG. If you're familiar with EEG arousal, the EEG is immediately activated. The animal's highly aroused, and I'll show you this video. There's no question the animal's aroused, because it's never seen a piece of chicken before. And you know, it looks aroused, but it's sort of anxious. And in that period, the cell is almost completely silent. The hypocretin cells are silent during anxiety, exactly what we found in the mice. But once the animal sort of relaxes and starts eating, all of a sudden the hypocretin cell goes up. But eating isn't required because the maximal rate we see is when the animals are exploring or when they're grooming. It's just these pleasurable activities, not particularly eating, and certainly not arousal in general. So this is implanted rat, and so he's asleep, and what you're hearing is the cell firing. And so clearly the rat is awake and the cell is completely off. So he's not too sure. I'll give an interpretation. He's not too sure that this is a good thing to eat, but as he starts to eat it, he seems to relax somewhat. And in fact, the cell starts firing as the EEG indicates a lower level of activation. Still awake, but a lower level of activation than during this initial period of anxiety. So 2 minutes later, he's decided, well, this is good. And he chows down, and the cell fires at this very high rate. So finally, we had the opportunity at UCLA to do a similar kind of investigation in humans. This is a very unique situation. Some humans with epilepsy can't be treated with drugs. I'm talking about epilepsy now, not narcolepsy. They continue to have seizures even when medicated, and this is obviously extremely debilitating. And it's possible to cure many of these patients by removing the epileptic focus. But it's very important that you don't remove tissue that's normal. And particularly if one side of the brain may be abnormal and the other side is normal, if you remove the normal tissue, then both sides will be abnormal and the effects will be devastating. So these patients are implanted bilaterally with electrodes in the skull, much as in the rat, although it's it's sort of more invasive in the humans because it has to go through this muscle at the side of the head. And the patients are then observed for the next month, and they pretty much are functioning normally. We're not interested, my interest is not in the epilepsy, and in between, most of these patients are not having any seizures during the time we're looking at them. In fact, we throw out any of that data. So we are able to put a microdialysis probe in the brain. So you're familiar with kidney dialysis where things are, a membrane is put in and the blood is filtered. Microdialysis, you can do exactly the same thing in the brain with, very little damage to the brain. So we're able to collect the hypocretin peptide while the patient engages in normal activities. And this is what we see. So here, this patient, when they're asleep, the hypocretin level is minimal. As you see, sleep is indicated there. When they interact with the physician, it goes up. In my experience, the physician is always telling them everything's great. Again, there's a gap there. Again, there's another interaction with the physician. It goes up. And then finally, a visit by family members, and it's the maximal level that you ever see in this patient. And so we did this sort of study across a bunch of patients, and we just published it earlier this year. And what we see in the column at the left is and we give these patients periodically forms to fill out how you feel happy, sad, hungry, thirsty, etc. And when the patient was saying they feel happy is when we see the highest levels of hypocretin, significantly higher than when they say they're unhappy. And the other thing I'd like to point out is right in the middle, the dark green, the last dark green bar shows when they're in pain. Because as I mentioned, these electrodes go through this muscle here, and sometimes these patients report pain, other times they don't. They are then treated with morphine, but we're not getting into that aspect now. But when they're reporting they're in pain, again, they're highly aroused, and the hypocretin level is actually at the same level as it is during sleep. So very low. Again, fitting with the animal data. So I'll summarize now what I think hypocretin is related to from these studies and some others that I don't have the time to discuss is positively motivated motor activity. We've seen this in the rat, as in the unit data you saw, in mice, the behavioral data that you saw. I haven't shown you the data from cats, but basically in cats it goes up during play. In dogs, it's high in play and interaction with their buddies. And in humans, it seems to be high at times of social interaction and particularly pleasure. I didn't get into this. Cataplexy is explained by the loss of hypocretin cells facilitating other neuronal systems, noradrenergic system and cells in the medial medulla. That's a whole other story, but we've published a lot on that, how muscle tone is controlled and how cataplexy results from the loss of hypocretin, but I don't really have time to go into that now. Now, several papers have reported that there's an increased incidence of depression in narcolepsy, and also in Parkinson's disease, where we've also found a depletion of hypocretin cells, although not as severe as in narcolepsy. And the fact that these cells are normally related to pleasure and that depression is high in narcolepsy and Parkinson's disease suggests that it's a relatively direct effect of loss of these cells rather than just all the other problems that result from this illness. And we think that the lack of the light-induced arousal in people with narcolepsy may also be due to this loss of hypocretin. So for us, a big message here is that arousal systems can be emotionally specific because, as I've shown, the hypocretin cell can be off and the people are highly aroused. In the case of humans or in the case of the mice. Well, something is keeping them awake. Is it histamine? Is it acetylcholine? Is it norepinephrine? This is what we're investigating, and I think we probably will have that answer soon, but it's a very different way of looking at arousal. In the past, neuroscientists have looked at arousal as sort of a common system, a lot of different inputs, and you're either aroused or you're not aroused. What this work shows is that there are certain kinds of arousal. There's arousal for pleasure, arousal to avoid pain, arousal for anxiety. It may be a very long list with all these different cell groups. So finally, I'd just like to acknowledge all the people who've worked on this. A lot of people have been involved in various aspects of this. Thank you very much.